cancer, who loses large part of the bone, all these people need a lot of substitute type of therapy. So, stem cell play a very important role, not only in this, even in skin, when though a small injury in your skin will heal automatically, but when there is a chronic injury like in severe burn, and in diabetic patients who have ischemic diseases, all these people, and people who have vision problem due to long time diabetes and hypertension etc. All these cases people are targeting stem cell therapy. So, why stem cell therapy? Those who do not know what is the definition of stem cell is, stem cell has the special property to proliferate excessively, extensively I must say, and large proliferation potential and its differentiation potential. So, what you can expect is, if you have the right stem cell, it can be differentiated into the cells of any of these tissues. That is the reason why people are targeting stem cell for regenerative applications. So, for long time, and for example, in 90s people were targeting embryonic stem cells, embryonic stem cells taken from the developing embryo on the embryonic day 6 to 8, the inner cell mass was taken out and it was cultured in the culture dishes using feeder cell layer and special medium etc. The advantage was large number of cells, generations after generation, up to 30-60 generations they could be proliferated, and large number of cells with similar properties could be obtained. But unfortunately, when people try to use embryonic stem cell for therapeutic application, major problem that they face is, because it is coming from allogenic origin, there is lot of immune reaction, and not only that, because they have the tendency to grow at a very fast rate. So, when it is implanted in animal models, it was found that there is teratoma formation. Teratoma is the growth and uncontrolled growth, and that was a major problem. So, the people are not able to use embryonic stem cell so much in the regenerative medicine. So, another stem cell has been developed by another group of two scientists, two Japanese people who have won the Nobel Prize for that they have developed a group stem cell type called induced pluripotent stem cell. So, what they have done, they have engineered the skin cell, from the adult skin, fibroblast was isolated, and they just changed 4 genes, and they were able to get stem cell known as induced pluripotent stem cell, which again everybody was very hopeful, thinking that okay, it is coming from somatic tissue, adult tissue, so there may be less problems, but unfortunately the induced pluripotent stem cell also behave the same way as embryonic stem cell. When it is implanted or transplanted in animal models, there is still teratoma formation. So, both ESC and iPSC behave similarly in terms of regenerative medicinal application. So, there is the only hope is to use adult stem cells. So, what are adult stem cells? Adult stem cells are stem cells which are left in each and every tissue at the developmental stage. So, when the embryo develops and tissues form, different organs form, each tissue is left with some stem cells. Example is skin stem cells. All of you should know that our skin regenerates on a day-to-day basis. The reason is because our skin, the topmost layer of our body, is exposed to different types of insults, like maybe it is UV rays or maybe environmental pollutions, chemicals. So, there is a problem of continuous damage to the cell. So, biology has planned it in such a way that the stem cells which are stored in the hair follicle bulge is involved in the regeneration of the epithelial layer, that is keratinocyte. So, day to day basis skin regenerates. So, another tissue which regenerates similarly is intestinal epithelium. So, intestinal epithelium also undergo lot of damage due to the food chemicals, and they probably for Indians, for us it is the spicy food, and all these are insulting the epithelial layer, and that also is regenerated on a day to day basis by the stem cells which are stored in the microvilli, that the stem cell pool. So, similarly every tissue. So, depending upon the regenerative potential of the liver also, liver is the detoxifying organ of our body. So, liver also come and undergo lot of toxic chemicals. So, liver also has the potency to regenerate in a faster mode. So, any tissue which has a faster regenerative potential, that tissue has large number of stem cells. And similarly, the one which are least regenerative are heart muscle and also the central nervous system, brain, but it does not mean that they do not have stem cells. They also have stem cells, but the number, the frequency is very low, therefore it grows, it regenerates at a very low rate, and especially when there is a big injury, like after heart attack, there is a myocardial infarction where the heart muscle is damaged in such a way. So, in that kind of condition, the stem cells which are resident in the heart muscle is not efficient enough to repair that. That is why regenerative medical applications are being explored by growing the stem cells in vitro and differentiating them into the right type of cell for regenerative medicine. So, if you look at the history of regeneration, so these are the how can we collect them, so when we want to use a stem cell for regenerative application, we have to consider from where it can be collected, which is the richest source which can be collected with least pain and least discomfort, and so many issues are being addressed. So, adipose tissue is one of the good sources for collecting mesenchymal stem cell. Bone marrow has large number of stem cells, and this stem cell from bone marrow is also released into the peripheral blood, and therefore some of the stem cells like hematopoietic stem cell and endothelial progenitor cell etc. can be collected from peripheral blood by collecting blood. And so, if you look at the history of stem cell therapy, bone marrow transplantation is the most successful stem cell therapy which is which has become a standard therapy. When I say it is a standard therapy, you do not have to worry about any ethical clearance. Any hospital who has the facility to collect stem cell from bone marrow or from peripheral blood can practice stem cell transplantation, and this is being used very routinely even in our RCC Thiruvananthapuram, and the success rate is very high to treat leukemia. More than 95% patients are cured by bone marrow transplantation, and the reason why this is being so successful is because the stem cell know the niche where to go. When you deliver it into the peripheral blood, it can recognize the same way the stem cells come out of the marrow and go into the peripheral circulation, the same way whatever is in the peripheral circulation can recognize the niche which is the bone marrow where the hematopoiesis process will take place. So, it is very successful. So, other than hematopoietic stem cell, there are other types of stem cells which can be considered for regenerative medicine. So, hematopoietic stem cell produces all kinds of blood cells in a routine day to day basis. Another one is endothelial progenitor cell. Endothelial progenitor cell is endothelium is the layer which is lining the blood vessel. So, endothelial progenitor cells, when you say progenitor cells, they are not stem cell, but they are lineage committed, they are lineage committed to vascular cell. So, the advantage is what is being known is that EPCs, generally known as EPCs, they also take part in day to day repair of the blood vessel, because every day there may be some blood vessel damage due to some small problems. Maybe you cut your vessel, or you crush your vessel, so there is a damage which you may not even know, but that is being repaired because the endothelial progenitor cells know, I mean that niche is comfortable for them to home and grow. So that is how the repair takes place. So endothelial progenitor cell has attracted many regenerative medical applications, because lot of diseases are cured, will be cured if angiogenesis is promoted. For example, in in diabetic ischemia, you must have seen lot of patients going around in the street with very bad leg ischemia because of chronic diabetes. The main problem is there is no angiogenesis. So to improve angiogenesis in that any ischemic tissue, whether it is after stroke, in the brain, or in the heart muscle after the heart attack. So people are trying to improve the angiogenesis by giving endothelial progenitor. So it has become a very important molecule. Then another one is multipotent adult progenitor cell, that are also released into the blood from the bone marrow, which can be used for the regenerative applications. Another fourth type is mesenchymal stem cells, that also attracts lot of work is being done, and only because bone marrow isolation is little painful, people have started using adipose tissue as a source for mesenchymal stem cell isolation. So I will be talking little bit about this endothelial progenitor cell and multipotent progenitor cells to demonstrate what we have done using confocal microscopy. Coming to the topic, so this much. So as I said, these endothelial cells participate in the repair of injured blood vessels. They can home to the site of injury from circulation. So we have started using this principle that it may be developed or it may be differentiated into endothelial cells to construct small diameter vascular grafts. Small diameter vascular grafts are nothing but blood vessels which are used. You must have heard about bypass surgery in heart. So the bypass surgery is done usually when coronary artery is blocked and there is no way to open it. So a surgeon will replace the blood vessel using a blood vessel graft which is taken from the autologous source like internal mammary artery or the saphenous vein. Most of the heart patient, heart surgery, after surgery you will see the patient also has a injury in the leg. That is because they harvest the vessel from that. So most of the time the patient is affected with lot of damage to the entire system. So therefore they are not surgeon is not able to get the right diameter vessel for replacement. In that condition, small diameter vascular graft is being developed using stem cell, which is differentiated into endothelial cell and smooth muscle cell. The lumen of the vessel will have endothelial cell, following which there will be smooth muscle cell, which gives the strength for the blood vessel to dilate and contract, and also it will be covered by fibroblast, which will give the strength to the blood vessel. So this is being engineered in various laboratories for developing an artificial though you can call it artificially developed, but it will have all the properties of the blood vessel because of the endothelial lining it will have. So what is done is it is seeded, the endothelial cell is seeded in the lumen, and the external ablumen, the smooth muscle cell is. So what we have tried is, because endothelial progenitor cell has the tendency to bind to the injured blood vessel. What happens in an injury? There is always a blood clot. When there is a blood clot, there will be fibrin formation, and fibrin formation and platelet activation will produce lot of growth factors which will participate in the wound healing process. So we tried to use the same principle by which the endothelial progenitors can be harvested from peripheral blood and grown on a fibrin matrix like this. It's a very thin layer of fibrin, and like this, and it will be included with various other molecules like growth factors and glycosaminoglycans to make a niche. And so the so we call it cell specific niche because we add growth factors which promote the endothelial differentiation that is being done. And also the another type of niche is also produced where platelet derived growth factor is added instead of endothelial growth factor to promote this multipotent adult progenitor cell to differentiate into smooth muscle cells. So, that is what I am going to show you. So, how is it done? It is the Japanese scientist Asahara et al. Has standardized this method of collection by density gradient or histopic centrifugation of diluted blood, and the buffy coat is collected which contains all the peripheral blood monocytes, mononuclear cells, and it is cultured, and small isolation is done in the culture to remove all the lymphocytes, and the monocyte fraction is put either in the EPC matrix or SMPC matrix, and then culture is continued with various defined medium. And then what you have to do, you have to prove that the differentiation takes place. That is where the confocal microscopy come into picture. So, in the culture what we have seen is within 4 days of culture there is a colony of cells. So, every stem cell will grow in colonies initially. So, a colony of cells, here you can see these are smooth muscles because it is more elongated even at day 4, and then because they are progenitors they have some stem cell property and they can proliferate, and by passage 2 they have shown the typical morphology, that is cobblestone morphology of endothelial cells and hill and valley morphology of SMPC. So, now we have to characterize, actually are they just from the morphology we cannot say these are the cell types. So, we use various markers. So, I talked about the matrix preparation. When you prepare matrix, why did you choose certain molecule? What happens if that particular molecule is not there? So, we have done a deletion study, like instead of adding gelatin we used only fibrin, or instead of adding fibrin and fibronectin we added just fibrin, instead of not giving growth factors we did not give growth factor, like that. So, this is an example. And so, vinculin is a molecule which is involved in the focal adhesion complex. So, we stain for vinculin. And so, when you have different studies deletion studies, we found that vinculin and vinculin expression and cell adhesion is affected when certain molecules are not there. That is how the niche is standardized. So, this is just to show that confocal microscopy played a role to show that vinculin expression is there, which is more in the presence of the composite of so many molecules which is added into the niche. Now, coming to the next part, you want to know whether the cell spreading, for any cell to be grown healthy, you need good cell adhesion and good anchorage and good spreading. So, we also checked whether the spreading is good by checking at the actin organization. So, when gelatin was not added, the number of cells were less, but on fibrin and fibronectin alone there is good spreading. But here when we added antibodies to fibronectin, that means fibronectin is blocked. When you add antibody it gets blocked, isn't it? So, so the spreading is very poor. So, whereas when you put all the matrix components together, you see all the cells are nicely spread and they are able to get a communication from the matrix, all the cells grow and differentiate because of the communication from the extracellular matrix. So, what we have tried to do is to develop a extracellular matrix composition in vitro in cell culture dishes to induce the differentiation, isn't it. So, this is the first level of demonstration that it is giving good adhesion and spreading. And next is the endothelial cell markers. One marker which can be used is 1 Willebrand factor. 1 Willebrand factor is one of the high molecular weight protein which is involved in the thrombotic process, and so usually it is found in one granule called ovable palette bodies. So, when you see this is just ordinary fluorescence microscope. The difference between this and this is confocal microscope. So, here what you see, lot of stray light and interference is there, and the granules are barely seen. In some cells it is seen, but in some cells it's just a green color. That is not acceptable for us. We want to prove that it is actually the green fluorescence is coming from the ovable palette bodies. So, when you look into the confocal microscopy, the same stained cells, you see that the granules are very prominent, and you can confirm, yes, these are one Lebrun factor. This is acetylated LDL uptake, which is another property of endothelial cells, which is going into the cytoplasm. Here also it is very blurred, and it is very not very convincing. So, when you look into the confocal microscope, what happened? No. Okay. So, the next one is I talked also about SMPC differentiation into SMC. We have so many other different markers, but I just to demonstrate the confocal part only I have used these slides. There are many other markers which are being used, like RT-PCR for endothelial nitric oxide expression, and PCAM, and so many other molecules are also expressed. Here in order to prove that the SMPs or the SMPs differentiate into SMCs, that is smooth muscle cells, we used markers, that is it is become white, sorry. So, that is the top left one is SMA, that is smooth muscle actin, and this is calpanin. So, both these are ordinary fluorescence microscopy pictures which are not very clear, but in the confocal microscopy the strands, the actin filaments are very clearly visible. So, this now this is about this is all about endothelial progenitor cell, which we use for construction of small diameter vascular graft by seeding into the lumen, and into the abdomen we seed this a smooth muscle cell and expose it to bioreactor, etc. I am not going into depth of that particular work. And another problem is wound healing problem. Stem cell biology is emerging very intensely in stem cell based therapy, because wound healing can be improved especially when skin graft from autologous source cannot be obtained from patients who undergo major burn injuries and large part of their body is injured. So, ideally autologous skin graft is the best treatment, but most of the patients cannot give from most of the patient you cannot harvest skin. So, stem cell based therapy development is a very important aspect in the research, and here also adult stem cells are being used, which is released from the bone marrow into the peripheral blood. These are called multipotent adult progenitor cell. So, same principle, the hypothesis we use is for wound epithelialization autologous PBMNC derived MAPC, that is multipotent adult progenitor cells may be harvested, and in vitro fibrin based biomimetic niche may be designed for selective isolation of keratinocyte progenitors from PBMNC and induce lineage commitment and proliferation, and a biodegradable polymer scaffold immobilized with fibrin and hyaluronic acid may be fabricated as a dermal substitute. So, whatever you saw in the earlier study, you must be seeing fibrin everywhere. Fibrin is just a matrix, just a polymer which will harvest other important molecules like fibronectin, which is very important in cell adhesion and spreading, and fibronectin is also capable of harvesting, serving as a very good reservoir for many of the growth factors. So, for each cell type you will decide which growth factor you want to use. Depending upon the cell type you need to get finally your growth factor composition will vary. So, the same principle of collection was used, and here we use for KPC niche we call fibrinogen epidermal growth factor hyaluronic acid, because our skin has a lot of hyaluronic acid which is important for growth of skin cells, then gelatin, fibronectin, bovine hypothalamus extract, then DME medium, etc. And finally, when we look at the cultures, in day 1 this is what you see, and by day 4 there is again colony formation. Lot of lymphocyte contamination is there. That is because the fibrin has a very good adhesive property, so non-specific binding also, but within few days the lymphocyte will get released and it can be removed by medium change. But whichever is specifically able to grow will grow, and this you can see the colony for becoming bigger and bigger, and by 15th day you can see the typical morphology of the keratinocytes in the culture. So, again we look at various markers to prove that using RT-PCR to show that. So, in addition to RT-PCR we also look at immunostained KPCs in 15 days. The markers used is P63, which is an epithelial marker, cytokeratin 5, which is a keratinocyte marker, and then fully differentiated cell will express a molecule called involucrin and filaggrin. So, all these proved that our culture system is able to differentiate stem cell starting from the PBMNC to keratinocyte. So, how do we use it in clinical condition? So, for that you need a support for to deliver, if the patient has got a 3rd degree burn and chronic wound, and that niche is completely destroyed, there is no area, there is no signal there, there is no extracellular matrix which can support the cell homing there. So, there are so many issues associated with stem cell therapy. So, what we planned is to have a biodegradable scaffold which is made of L-lactate and L-galactate, because they are considered to be degradable within 2 to 3 months time, and they produce L-lactate and lactic acid and glycolic acid, and that is normally found in the human system, and it gets cleared. So, therefore, this polymer is approved by FDA for human use, it is non-toxic. So, that is why, so this we made a scaffold by electrospinning technique, and then this is now the polymer has no adhesion site for cells. So, you need to improve the adhesive property of the synthetic polymer. So, for which again we coated this polymer with fibrin, because fibrin is a natural scaffold in all wound healing processes and cell can adhere and grow. So, then that we call as hybrid scaffold. So, this is the bare scaffold made of synthetic polymer, and hybridized with fibrin it becomes hybrid scaffold. So, when you culture adipose derived mesenchymal stem cell, why adipose derived mesenchymal stem cell is because it can produce the dermal tissue easily, it can differentiate into dermal fibroblasts with very little effort. So, if ADMSE can be seeded on this hybrid scaffold, once dermal skin, dermal part of the skin is developed, it is easy for the epidermal part to grow. So, without dermal support epidermis cannot grow. That is the reason why we first started growing ADMSE on the scaffold. So, after 30 days of culturing, if you look at the ordinary stained microscopy, you will see a lot of cells, but you do not know whether, because the scaffold is thick, whether the cells have migrated into the scaffold and is it possible, or is it in the superficial region, that you have to analyze. Only by using confocal microscopy you can study that. Even the ordinary fluorescence microscopy can only give a picture of the superficial part of the scaffold, that cells are aligned there. That is what you see here. You can see barely the fibers here. So, again in vitro construction of dermal using degradable scaffold, if you compare bare scaffold with hybrid scaffold. So, bare scaffold I said, because it does not have lot of adhesive sites, the number of cells are less, and and some of them are autofluorescence, because this is ordinary microscopy, and hybrid scaffold there are more number of cells. So, the scaffold, when you have to analyze with confocal microscope, because you can give you can select a different color for the scaffold fibrils and a different color for cells. So, what this red fluorescence that you see are of the polymer, that is PLGC, and then the green ones are the cells. So, then you can combine both and produce a combined picture. So, when you grow these cells for 10 days on the scaffold, this is what you see. The cell is already there and it is penetrating into the scaffold, because scaffold is porous, and that is a very good sign, because that cells are able to migrate into the scaffold and grow and form a good tissue. So, now if you look at it on 20 days, the same culture after 20 days. So, here the green color is the polymer scaffold or the hybrid scaffold, and the red is the cell, and then the combined picture is seen here. And after 20 days, when you look at the merged picture, you will see very hardly any green color. That means cell is populating the scaffold very nicely by 20 days. Now, look at it in 10 days, I mean after 30 days. Green, it does not mean that scaffold is gone. Scaffold is there, but the cells have completely covered the fibrils, and there is a very good sign, that is a very good sign that the skin, good skin formation can take place. So, then we look at the actin also, whether whether the after this sectioning is done to find out whether in all layers this cell has penetrated, which you can see because all of you have undergone the training, what it means, in 3 micrometer thickness the scanning was done, and it is the combined picture. So, now skin, when only cells growing in the scaffold does not make a skin. It has to, if you have to get the suppleness of the skin, it has to, the cells have to synthesize extracellular matrix and deposit on the skin. That is when regeneration, you can call regeneration only when the extracellular matrix is also produced, because your scaffold is going to be degrade in 30 days, in 30 to 60 days the scaffold will go away. So, what will support the cell is the collagen and the elastin that will be deposited by the cell. So, this was experimented by decellularizing the scaffold. I showed you 30 days we have grown the cell, and after 30 days we have taken a cycle of decellularization process by treating with various detergents and washing it and etc. And what remains is only the extracellular matrix, and which is stained with collagen. So, this is the green color that you see is the collagen. So, you can see the network of collagen deposited in the matrix even after the cells are removed. So, the green here is the scaffold, blue here is the collagen, and the merged picture you can see down. So, most of the scaffold is covered with the collagen. So, this is elastin, this is stain for elastin. So, as compared to collagen, elastin content is much less. See, it is very obvious from this picture, because green color is seen, that means the scaffold in the PLGC is showing up, and still it is a good sign that elastin is there. So, that the regenerated skin will be elastic in nature. And we also did a study to see whether 2 layers of skin can be generated in vitro. For that, on the fibroblast, the differentiated the keratinocyte was seeded, and to show that it remains there in the fibroblast layer, we did staining of cytokeratin 5, and you can see the fibroblast layer and the cytokeratin. So, this is another staining. So, that way to conclude, confocal microscope has been a great tool in stem cell research leading to tissue generation, that to locate protein to prove differentiation to various lineages, and to prove the effect of various matrix protein in focal adhesion and signaling, and to prove the effect of matrix proteins in spreading, and to prove cells migrate to pores and populate the biodegradable scaffold, and to prove ECM deposition such as collagen and elastin after long term of culture on the scaffold, and to finally prove cells survive in differentiated state upon co-culture. The last one which I showed is a co-culture. So, thank you for your patient listening. This is my laboratory where my students who have done lot of work, just whoever was involved in this PBMNC culture only I have. Thank you, sir. If you have any questions, I am happy to answer the question. Nobody has question? Yeah.
So the extracellular matrix you see put down by your stem cells, is it very similar in concentration if you examined it versus just what would be done like in a native vessel? Is it very similar in composition?
Yeah, it is very similar. Because we select the collagen type depending upon what tissue we are growing. So, we also look for negative staining, like all types of collagen we stain to make sure that we get the right type of collagen. And not only that, by doing real time PCR, we make sure that this collagen deposition cannot be lifelong, then you will get a very fibrous tissue. So, what we have found was using real time PCR, we could find out that by 17 days there is down regulation, or the quiescence has reached. So, that shows that there is no continuous deposition of collagen, it will be regulated at time. So, this is actually my student Ranjit, he has done animal studies in rabbits, in diabetic rabbits, and he has got very good result, and which has been published, and he found there is angiogenesis, because he mixed keratinocytes and EPCs together when he transplanted with the so
angiogenesis is as good as new, basically.
Yes, it is as good as new, because fibroblast will not synthesize elastin. That was surprising. Usually after 20 years of our age, we do not synthesize, our skin fibroblasts will not synthesize. That is why when we have a big burn injury and it heals, but it does not become elastic, you can see that there is a scar formation, which was a different feeling when we had inference when we used stem cells. Stem cells are able to the quantity is not as much as elastin. It was quantified also using commercially available reagent kit, how much collagen per centimeter square of scaffold, etc. It's very extensively studied, but for this conference I just picked few slides which has relevance here.
So is there a big difference in the age of the stem cell donor? Do you see a big difference, like
there is a lot,
yeah,
yes, we find lot of difference, especially when we collect ADMSC. Ours is a hospital where the cardiac surgery and neurosurgery is going on, so we collect adipose tissue from patients who undergo cardiac surgery, because that was accessible for us, it was easier for us to get ethics committee approval, so we collected. But what we found was all of them are patients with different age group. Some of them are 70s, in their 70s, some of them are very diabetic, some of them have other hypertension. So it was very difficult for us to get donor to donor similarity, repeatability. That is important when we do research. When we have to publish, we have to show repeatability, which was not possible. So we started collecting from young donors who undergo lipoaspiration, who is healthy, so that when we collect such lipoaspirates and isolate the stem cell, it does not mean that these older people do not have stem cells. They have stem cell, but their proliferation potential is much reduced. In 3 to 4 passages, they are taking more time, maybe 4 days to passage. So whereas in lipoaspirates, every 3 days you can passage and multiply cells and differentiate, and we look at trilineage differentiation into adipo, chondro and osteo. So that also comes out very easily when we collect from young individuals who electively go for lipoaspiration because of cosmetic reasons. Older patients have problems.
So I guess it makes sense, younger and healthier just works better.
Works better, yeah. But the advantage is ADMSC is considered to be safe for allogenic transplantation also. People think it is okay, it is less immunogenic, there is no MHC class 2 antigen. So I am not sure, I have not tested. That is what the literature says, that it is okay to use allogenic. So in that case, lipoaspirates from clinics can be collected and differentiated into any type of cells, and it can be used. But flow cytometry and confocal microscopy, RT-PCR, these are our important tools, and we have a Zeiss machine in our institute, and so we are relying on that.
Very interesting.
Thank you.
Madam, at this time, almost all this maternity hospital, like at the time of childbirth, they say that to collect the sample for the baby. What is your opinion on that?
It's an unproven expectation, so I don't have any high opinion about it, because they are spending lot of money to collect the code and store it and maintain it. Every year you have to pay some money, and you are anticipating after 30 years the child will have some disease, and they will use it. So at that time, whether it will be whether we can revive it and get the same, like the question he asked, after aging, so after storage what happens, after long term storage, though they put it in minus 175, that is in liquid nitrogen, and but it's somebody has to prove it will work. Till then I don't advocate on that. It's a waste of money. You can as well go for allogenic ADMSC, which are being sold by many companies. It will have the same effect, or else autologous. Many when the technology is proven and standardized, many diseases can be treated using autologous ADMSCs. All of us have enough fat to collect ADMSC, so fat will become a blessing after some time for collection of ADMS.